Hi everyone. Now in this part we are going to discuss about mechanism of enzyme action, which is one of the part of this enzyme topic. Okay, let's begin. So mechanism of enzyme action, which can also be called as biocatalysis. Why we also call them as biocatalysis means enzyme is also called as biocatalyst. So as we know, enzymes are proteins which tremendously increase the efficiency of rate of biochemical reactions at normal cellular conditions. Yes, as they are participating in the biochemical reactions and catalyzing the reaction, we call them as biocatalyst also. Then what is a substrate? The molecules upon which an enzyme acts are called its substrate. For example, this is a substrate. That means these enzymes are going to act on these things, molecules. So those are being called as substrates or substrate molecules. Then what is the active site? So the active site is a complex 3D cavity, okay, into which this substrate molecule fits is going to be termed as active site. And at the active site, the enzyme works by lowering the energy of activation, all the things we'll discuss now. Okay, so the active site is a complex 3D cavity into which the substrate molecule fits. Then in 1913, Michaels and Menton proposed that an intermediate complex is going to be formed during this enzyme activity. What is the enzyme activity that it is doing? This enzyme and substrate combine together and giving rise to the enzyme plus product. So that is what we know, isn't it? Now they've explained or proposed that there will be an intermediate compound that is formed between the formation of a product and the substrate. So what is that called as enzyme substrate complex? So the binding action of these, both the enzymes and substrate, forms a transitional or intermediate compound, as we are calling it as enzyme substrate complex, that was proposed by Michael and Menton. And one more thing that you have to remember here is enzymes promote a given reaction but itself remain unchanged at end of the reaction. See here how the enzyme when combining with the substrate after the product formation also it is going to be the same. So that's how it is going to remain unchanged at the end of the reaction, and such that this enzyme again combines with another molecule of substrate, and this is going to continue. And for this binding action, these two are going to have some sort of a bonding, and that binding action makes both the substrate and enzymes stable. Okay, the contact between the substratum and enzyme is going to be either by ionic bonds or by hydrogen bonds or by van der Waals forces. Now, this active site, as I told you, the active site of an enzyme has some special groups such as a amide group or carboxyl group or sulfur hydroxyl group. As it is a protein, so obviously it is going to have these groups which bind to the substratum through the above bonds, as I told you, the hydrogen ion or van der Waals forces, to form a transitional intermediate. Okay, so what is the transitional intermediate? Nothing but our enzyme substrate complex. Okay, so that's all because of this bonding. So what kind of the bonds will be present between the substratum and the enzyme? Maybe hydrogen bonds or maybe ionic bonds or van der Waals forces. So these bondings are going to have a reaction with the type of functional groups that are present in the enzyme active site. So that's how you are going to have the formation of an intermediate transitional state called as enzyme substrate complex. So now what is happening? So this reaction, that means the reaction between the enzyme and the substrate, is an exothermic reaction and releases some energy. So when they are being put together, that means when they are combining, that is substrate and the enzyme are coming together, they are going to release some sort of energy that is going to be called as exothermic. So the reaction between the enzyme and substrate is going to be considered as exothermic and releases some energy. So with this energy, what happens? So rises the energy level of the substratum. So now the reactants are so dull, so that means the substrates are so dull, that means will do the work so slowly. But once the enzymes comes and binds to it, and they are going to activate this reactant such that they can be converted into products very easily, and that energy is being called as activation energy. So energy released will rise the energy level of the substrate. This activating phenomena is being termed as activation energy or energy activation, which is being possessed by this substrate molecules. Remember, the activation energy is being gained by the substrate molecules or the reactants. Okay, then what is the activation energy? So how can we define this activation energy? For each and every reaction to occur, a minimum energy is required. So for every reaction of the conversion of reactants into products or substrates into products, there should be a minimum energy should be required. And that energy is called as activation energy. So if the reactants want to convert into the products, they require some sort of energy, and that energy is being called as activation energy. And for each and every reaction to occur, a minimum energy is required. That energy required is called as activation energy. An enzyme, what the enzyme is doing. For example, without enzyme, and here I have given you the with enzyme. So without enzyme, these reactants have to have this much of the energy to get converted into the products. And obviously the distance is more. For example, if you want to reach from one place to another place and you are so dull, or by walking you are going, so what happens, it takes a lots of time to reach your destiny. So but for example, if you are having a lift, for example, of any car or any vehicle, so obviously what happens, you are going to be converted into the you are going to reach your destiny within less time. So your activation energy, without spending the much energy, you are going to reach your destiny. So in the same manner, here the reactants or the substrate molecules without the enzyme requires more activation energy. So this is the activation energy without the enzyme. But in the presence of the enzyme, these activation energy is going to be lower. That means reaching is going to be very easy. That means the conversion of substrates or reactants into the products is getting very easy because of lowering the activation energy in the presence of enzyme. Okay, so as enzymes lower down or decrease the activation energy, by which the rate of reaction automatically what happens, increases. The rate of reaction is going to be increased. So that's how your enzyme plus substrate is going to form the enzyme substrate complex, and these are giving rise to your final enzyme plus products. When an enzyme and substrate are binded, so one more thing that I want to tell you here is, this is all about the activation energy. When enzyme and substrate are getting together, this enzyme is going to release some sort of a energy or liberating some sort of energy. Enzyme, I'm talking about the enzyme. Previously I talked about the activation energy is related to the reactants, okay, and now I'm going to put a note on the enzyme releasing energy. That enzyme releasing energy is called as binded energy. That means when an enzyme and substrate are binded, the enzyme or the energy, whatever the energy, enzyme or substrate, the energy that is released or liberated is going to be called as binded energy. And this binded energy is used to lower down the activation energy of the reaction. So when a substrate and the enzymes are going to be binded, there will be a release of some binded energy. And this binded energy is going to lower our activation energy. So that's how this enzyme substrate complex is going to be formed, and it is lowering the activation energy and finally giving very easily this enzyme and products. And where, as I told you, this enzyme, it is not going to change and it can be retain its shape of at the end of the reaction. Okay, now this mechanism of enzyme is going to be proposed to explain enzyme substrate formation. So how many mechanisms are going to be there to explain about this enzyme substrate complexes? Two. Okay, so there are two mechanisms have been proposed to explain the substrate enzyme complex formation, that is EC ESC complex or enzyme substrate complex formation. So what are those two? One is the Lock&Key model, another one is the induced fit theory. Okay, so let's see the first one, Lock&Key model. So Lock&Key model was proposed by Emil Fischer. Okay, and according to this theory, the reaction of substrate and enzyme is analogous to Lock&Key model. So the reaction between the substrate and this enzyme is going to be resembling the lock and key. For example, here you can see the key and this is a lock. These are going to be specific to one another. We can't open another lock with some other kind of the key. That should be specific. In the same manner, the enzyme and the substrate should be specific to convert and form the product. Okay, so enzyme is analogous to key, as I have told you here. The enzyme is analogous to key. Sorry, here it's a lock is an enzyme, and here you are going to have the key as a substrate. And key model, Lock&Key model, the geometrical configuration of the socket is fixed. That we know that lock is going to have the fixed shape, and similarly the substrate is analogous to key, so obviously whose configuration is also fixed. Hence the enzyme substrate complex is assumed as a rigid lock and key union between the two. So here, if this is the shape of the substrate, that kind of the active site should the enzyme possess. Then only we'll have the formation of enzyme substrate complex, and it is going to have the enzyme product complex. Finally, it is going to be converted into product. If this is not the key, some other is going to be there as a substrate that which is not relevant to this enzyme, then obviously it is not going to form the product. Okay, so that's how, as this is going to be resembling or analogous to Lock&Key, so obviously this was called as Lock&Key model. But the portion of enzyme to which the substrate combines as it undergoes the conversion to a product is called as active site. So there is some sort of a drawback. What is the drawback of this Lock&Key model is, if the active site were rigid and specific for a given substrate like Lock&Key, the reversibility of the reaction would not occur, isn't it? So if you want to separate the substrate and the enzyme, so we cannot do it, but we are able to do it, no? So why this is not this is going to occur means, so that shows that this active site is not a rigid one, it is flexible. And to explain this drawback, there is another theory called as induced fit theory, which was proposed by Coastland. So let's see that one. Okay, so as I told you, lock is going to be enzyme and the key is going to be your substrate. So induced fit theory, as I told you, that was proposed by Mr. Daniel E. Coastland. Okay, and this induced fit model is a modification to the Lock&Key model and is generally thought to be the more accurate version. When we compare to the Lock&Key model, this is the more accurate version. And according to this theory, the active site is flexible. Whereas in the Lock&Key model, I told that the active site is going to have the rigid form. That means it won't change. The geometrical configuration of active sites of enzyme is inducible. That means we can modify that, not we. So this enzyme active site is going to have the modifications while it is going to have the combined with the substrate. The enzyme active site changes configuration such that it can accommodate the substrate better. Okay, so all enzymes, as I told you, is having an active site where this enzyme is going to bind, which is specific to this enzyme. When the substrate enters the active site of the enzyme, see here the enzyme is slightly changing its shape very slightly to accommodate this substrate better. And this is called as the induced fit model of enzyme action. Induced fit model of enzyme action. Now the substrate once enters this active site, the enzyme slightly changes its shape to accept the substrate as an enzyme substrate complex is formed, isn't it? Now a substrate is coming and this enzyme is going to change its shape slightly to accommodate this substrate. Once it has come, it is going to form an enzyme substrate complex. Now this substrate is going to be converted into the products form, and the products exist. That means they are going to be released from the active site, upon which point the enzyme returns to its original shape, and it is going to be back to its normal shape. So this is what the induced fit theory model is going to tell. And there is a little bit extension of this topic that I will tell you, that this active site is going to be of having two sites. So these two are active sites only, but they are going to be classified into two sites. So one is going to be called as buttressing group, another one is called as catalytic part or catalytic group. That means the active site of an enzyme is going to possess the two parts. One is called as buttressing group, another one is called as catalytic group. What is this buttressing group does and what is this catalytic part is doing? The buttressing part supports the enzyme to bind to it. So here you can see the shape of this substrate, and here you can see the enzyme's shape. So it's quite different, isn't it? But this enzyme is going to have the supporting or changing its shape such that the substrate can fit into it. So here the buttressing group supports the substratum. That means it will hold the substratum properly such that it can bind clearly. And the catalytic part, what this catalytic part is doing, it is breaking the bonds between the enzyme and the substrate such that it can form the products. So that's how both parts of the active site are going to be very important in the conversion, in the binding of the substrate, and as well as to the conversion of the substrate into the products. So this is all about the induced effect theory. And out of these two theories, that is, what are the two theories that we have discussed? One is the Lock&Key model and another one is the induced effect theory. Okay. Induced effect theory is more acceptable. The reason, as I told you, it explains the broader group specificity of enzyme catalyzer reaction. So out of these two, this is the one which more acceptable. So as we know, the reasons is broad specificity enzyme catalyzed reaction. So here, if once again in the Lock&Key model, you are going to have the active site as a prefix and the substrate that is going to fit only, then only you are going to have the formation of enzyme substrate complex. Otherwise there is no reason, as these two are going to have the analogous to a lock and a key. So this model is going to be called as Lock&Key model, which was proposed by Fischer. And then coming to the induced effect theory. So this induced effect theory was proposed by Coastland, and here according to the Coastland, the active site is not going to be fixed. What it is happen? It is going to have the flexibility. The active site of enzyme is flexible according to the substrate to fit, and this active site is having the two parts. One is the buttressing part, another one is going to be the catalytic part. The buttressing part is going to support the substratum to bind with the enzyme, and the catalytic part, after the formation of the enzyme substrate complex, the catalytic part is going to break the bonds and conformational changes to convert into the products. So that's how the mechanism of enzymes with two theories explanation we finish in this part. Okay, thank you.